Hello and good afternoon, viewers. This is Dr. Ali Atiq one more time with another interesting webinar series. Before we embark to our webinar, like always, I want to give a brief description about the faculty. I have told you about myself earlier, but one more time. What's the harm in it? I'm Dr. Ali Atiq, and I am the head of Department of Pharmacology and Toxicology. And just like me, there are other five departments. When together we make Faculty of Pharmacy. We have five programs being offered in the Faculty of Pharmacy right now, starting all the way from the degree to all the way to the postgraduate PhD program. Our diploma program is the first and the foremost. I will discuss that. It's a 3-year program, and with the completion of this program, you will be by the end of this program. You will be called as a pharmacy assistant, and it has a very important role in the society and also in the pharmacy profession as well. The second one is our is the Bachelor's of Pharmacy program, and it's a 4-year program. And the end of it, you have to undergo the PRP or the clerkship for 1 year. But after that, the title will be the pharmacist. And I don't need to emphasize what a pharmacist has its impact on the society. Then we have a newer program with that, which is Bachelor's in Science in Pharmaceutical Technology. Now, with the pandemic is ongoing, you know that the pharmaceutical industry has worked robustly in order to produce the vaccinations and the other solution against this menace of this virus, which has changed our life completely. So by the completion of this 3-year degree, you'll be able to embark your profession in any industry, from pharmaceutical to industrial and all the way to the cosmetic as well. Then last but not least comes our master's and PhD program, and these are research based. And after completing this, you can be part of the big research institutes around the world. Now, without any further ado, let me introduce a little briefly about our today's speaker. Today's speaker is Miss Chong Wei Sa, and she has done her graduation from University of Malaya, and she is done her master's in instrument analytical chemistry, and she's formerly working in the analytical chemist and the commercial analytical laboratories as a QC chemist. And the experience is quite wide with the OEM lubricant oil manufacturers and application support in Thermo Fisher Scientific. And I have used their product during my PhD as well, so I'm quite aware of the Thermo Fisher research heritage. And she is an experience operating and supporting analytical instrumentation, for example, GC, HPLC, UV, and many other scientific instruments. And today we have her with us. So without further ado, I would like to call Dr. Chong Wei-Sa. The floor is all yours, ma'am, and please give us the knowledge about today's topic. Thank you so much.
Okay, thank you, Dr. Ali. So I'm Chong Wei-Sa from Quaternary. You may call me Chong for short. So today's topic, I would like to share the principle of HPLC and the application in pharmaceutical analysis. So first of all, Quaternary is a US-based company providing many solutions in the laboratory environment, okay, for all variations of industry, from the inorganic, organic, or the spectroscopy's perspective. So this is the demo lab at our office in Petaling Jaya, not far away from your campus. So if you have a chance, we would like to extend the invitation one day, okay, to visit our classroom training in depth for different instrumentation. So first of all, to start with the topic today, we would like to explain on what is chromatography about and how did it start at the beginning. So it was discovered by a botanist rather than a chemist. From the botanical research, by using the plant and paper chromatography. And the word chromatography is actually from Greek roots of word, means to write or record the colors. Okay, so this technique is involving the different rate of movement of the solution through the stationary phase or any phase that is not moving, but at the same time, the analyte or the target compound will be bringing upward or to the destination by the solvent or mobile phase. We will be discussing more about this later. But in the old days, you may try this in school where the teachers has given us a piece of filter paper and we are asked to crunch a piece of leaf out there from the school trees, okay, and we have to drop a juice extract onto the filter paper and put it into the test tubes. The test tube with some solvent inside, probably hexane or toluene, and with a dip of this plant-based extract on the filter paper, the solvent will then move the compounds or the plant extract upwards. And you can see different colors appears on the filter paper. And later on, these have been doing the similar way to the gel type of stationary phase, where a gel type silica phase being placed in the tubes, and we pour the plant extract into the tubes to let it flow by the gravity. So upon the time pass, we can see the different pigments with different colors eluted or flow out by gravities at different time. So there are some pigments that is flowing out at first, and some other colors of pigment, in this case the blue, will be flowing or eluted later at different time because they have different properties. Some other example of the chromatography would be the liquid chromatography, and you can see just now the paper chromatography, and this technique has been applied to the thin layer chromatography as well. Okay, and the last one will be the gas chromatography, in where rather than the solvent bringing the plant extract to move towards the stationary phase, and this time we are using the gas to push the analytes to the detector end. So in old or classic column chromatography, this is what we explained just now. Okay, we have two phases where the solvent that can bring over the sample or analytes will be the mobile phase, where this phase is moving all time in the system. At the other hand, we have the stationary phase, where in this case, it could be a silica gel in a tube, and we will inject the sample at first, followed by flowing the mobile phase, and let the mobile phase to carry the analytes from beginning until the end. And this is kind of the separation. By having the different absorption or affinity of different compounds in the mixture, we create different time of each compound to reach the test tube for the collection. So this is the separation technique in old days, and it could be used for purifying process, because upon the separation done, it is also means that we are collecting the pure compounds from the mixture from the beginning. So in these slides, we would like to introduce two important concepts in chromatography. Okay, the first one is the stationary phase, which is not moving at all, and the other one is the mobile phase, which is moving all the time. Okay, so this concept is using the gravity in old days, but for the modern technology, we are using a pump rather than the gravity. And the column to replace the silica gel those old days are now packed with a smaller particles and coupled with variety of online detectors. So this technique combined together, we call it liquid chromatography. And this is the modern instrumentation rather than the gravity-based system. So what is liquid chromatography? It is actually a separation method that relies on the differences in absorption or affinity based on the behavior between the analyte in the sample with the mobile phase and stationary phase. So as explained earlier, mobile phase is the solvent that could probably dissolve your sample and move all the time in the system. But the stationary phase is a phase that adhere to your sample analyte temporarily but not moving at all. So this is the sketch of the HPLC system. We have the solvent here as mobile phases. It probably more than one solvents because we can always mix each one of them to create a different composition in different solvents to be bring over to the entire system by the pump. So at the same time, the mobile phases or solvents would be flowing in the system all time non-stop. And when we inject the sample into the HPLC, the carrier or the mobile phase will be carrying the analytes into the column. And here is where the stationary phase placed and separation happens. Finally, depend on the affinity of components to the column absorption. Some compound will stay in the column just for a little while, and some other compound have higher affinity to the column phase or stationary phase will stay a little longer, and each compound will reach the detector at different time, and the signal will be processed by the data system and being used in the different application fields. So there are generally two types of chromatography techniques nowadays. One with the gas as the mobile phase, another one with the liquid as a mobile phase. So these are the difference for the GC gas chromatography. For if you have solid sample, then we might need to extract something that is interested for separation by the headspace unit or solvent extraction. And if you have the liquid sample, maybe we can do the direct extraction. And for the gas samples, for example, the gas monitoring, we could inject the gas directly or collect the gas by some other absorbents and finally release into the GC. As for the liquid chromatography, some of the solid samples could be extracted by the similar polarity of solvents and then inject into the HPLC. And some of the liquid samples, after filtration, simple filtration, could be directly injected into the HPLC, depend on situation. But unfortunately, for the gas samples, it is not suitable for the liquid chromatography technique. So when you choose the technique, probably I hope this slide would bring some idea to you on what kind of samples is suitable for LC or GC. So same thing, the mobile phase is the main difference of both techniques, where we are using gas as a mobile phase for GC and solvent for the LC depends on what are the modes of LC operation. We will discuss this later. So how is HPLC separate our mixture? So it operates in a few modes. Generally, this is normal phase, reverse phase, and ion exchange phase. Of course, there are many other phases like size exclusion is not explained here. Okay, so in the normal phase is what the people start earlier, where they are using some organic solvents such as the hexane, toluene, or benzene as a mobile phase. And of course, these are the solvent which is not the polar phase. As a reverse, the stationary phase or column has to be polar, which is reverse to the mobile phase itself. So this idea is because the mobile phase should hold the analytes of interest less than the stationary phase. So if we have the polar compound, then we have to use the polar column or polar stationary phase. But in these days, reverse phase is the most popular in use because most of the analysis is done in the reverse phase, where the mobile phase we use is a polar, for example, water, methanol, acetonitrile. And we can mix few reagents together or solvents together to create different polarity situation so that different compound with different polarity affinity will be eluted at different time. At the same time, the stationary phase or the column would be nonpolar because reverse phase is targeting for nonpolar compound as well. So if you have ever operate HPLC, probably C18 column is the most that you have been using. So this is the reverse phase where you are using C18 column, which is the nonpolar column, and at the same time, you are using some polar mobile phase to carry the analytes to the detector. So the other mode is exchange, ion exchange, where some buffer will be involved. So this would be involving cation or anion exchange during the separation mechanism. So this is the overview of GC versus HPLC. Okay, you need a gas tank, purified gas for the GC. Okay, and then inject. Same, you have injectors in the liquid chromatography as well, but rather than a gas tank, we are using the solvent and we need to degas to remove the air or dissolve air in the solvents, then being pumped to the entire system. And also we have different forms of columns here for different techniques. And finally, this column is attaching with various detectors for the GC and HPLC. So this is what the chromatogram look like. This is the results from the instrument, whether GC or LC. So basically, from a mixture, we hope different components now is hitting the detector at different time so that we could separate the mixture into individual peaks. Here, this is what we call chromatogram, where the y-axis is the unit of detector itself. It's not necessarily absorbance. It depends on what kind of detector are you using. And the x-axis is always the time in minutes because we are measuring what is the time that for each components to reach the detectors. So these are the column used and the mobile phase that used to separate these compounds. Okay, and you can see it involves flow rate and pressure because we are no longer using the gravity to separate the components, but we pump the mobile phase to entire system with a fixed flow rate. When we are talking the flow rate means how fast the mobile phase or solvent is flowing in the HPLC system. And this is what we call flow rate. And when we involve the flow rate, we also using the pump to generate the flow rate. So here you can see the pressure involved. And finally, the samples, how much the samples being injected into the systems. So this slides illustrating how the chromatography process is done. So at the beginning, we have a mixture from the samples, whether we treat the samples of solids or liquids or direct injection. You can assume all the compounds in your sample vials is a mixture. And then by using the polarity difference and also the some different composition of solvent to generate different environment because each components here having different affinity to elute or the rate to hit the detector. So how do we separate it? We are using the characteristic of different affinity of each compound to the column material itself, so that we generate different time of each compounds to reach the detectors. And after the separation has done, we got the results or raw data that we say this is chromatography, and every peaks representing one compound or one organic molecule. So we are hoping that all of them is separating at different affinity to the column or stationary phase, so that they reach the detector at different time based on the characteristic of absorption and polarity. It could be more factors, but these two are the most common. So chromatography technique could be used for qualification and also quantification. So at the first, how do we quantify or identify the unknown? What is containing inside my sample? So first, we need to inject the standard test mix that you might have some targeted compound in your mind. Okay. After that, this certified standard mix and under the same condition of chromatography being tested. Okay. And we record each standard with different retention time, because when you inject the standard test mix, you are given a certificate of analysis. When you purchase a standard mix, it already well defined what is eluting the first and the last. So after you have done this, you determine different retention time for each component here. Retention time means the time for the component to reach the detector. It will stay in the column for a while and leave the column and reach the detector. So after we get this chromatogram from the standard test mix, we also inject the unknown samples where treated earlier, and then we compare the chromatogram of the samples against the standard test mix. And now we know if both having the same retention time, and we can say the first eluted peak is dicing in the samples. So this process is going through the whole mechanism of injection, okay, where the mobile phase flowing all time in the system. And when the sample mixture arrive in the column or separation phase, stationary phase, separation takes place, and each one of them will reach the detector for detection. So it is not really the unknown detection by just injecting the sample into the HPLC. You must have some knowledge to your sample. In your case, probably the pharmaceutical products. And what mean by not really the fully unknown determination? It is because we still need to compare against the standard test mix where we purchase from a certification body, and compare against the retention time. So this is particularly useful in the production of pharmaceutical products rather than biological samples. So after that, first, we are using the chromatography system or HPLC to generate the peaks. And then we have to prepare different concentration of test mix, because at this moment we only identify the identity of each peaks, but the next question to reach us is how much of each of them. So to quantify them, we need to prepare different concentration in series of each solution and mix into one sample vials, and then from the low concentration we inject until the higher concentration. In this case, we calculate the area of the peak for each component here, the area of each component, every compounds. And then let's say from 10 ppm to 1000 ppm. So you might prepare 3 to 5 within the range, let's say 10 ppm, 100 ppm, and 1000 ppm, for example, and you inject into the HPLC system. The system will calculate the area of the peak and generate a calibration curve for the standard that you prepare. And finally, tabulate the linearity of the calibration curve and the R square. Okay, the higher the better. And then after establish the calibration curve, we can now inject the unknown samples that is treated the same way as the standard do. And we also calculate the area as the standard do. To quantify the area of unknown by using the calibration curve here, normally the response of area would be the y-axis, and the concentration would be the x-axis. So by doing this, okay, quantify the area and read the concentration at x-axis, and we can now quantify what is the concentration or amount for the compound of interest in the unknown samples. So who would need the separation to be done, and why do you need this HPLC or GC? It is to separate the mixture of compound, and when the separation takes place, at the same time, you actually doing the purification, and this technique could be used to identify the unknown. Of course, with the non-standard injected, then after comparison, you might conclude the unknown samples. So this technique is particularly very useful in pharmaceutical for the bulk analysis of raw materials, for the finished products after manufacturing, for the environmental testing or food safety, and it could be applied to wide range of application, mainly for the formulation purpose. And when you formulate pharma products or tablets, probably you also need to check the impurities, okay, assay in the formulation, and also as quality control. And finally, of course, the compliance requirement before selling to the bulk market or global market. So we have been sharing the principles of chromatography. So there are a few ideas that we should learn towards the presentation just now. The first thing is the chromatography technique is for the separation of the mixture in the compounds. And in this separation technique, there are two major terms that we need to learn. First is the mobile phase, which is like a carrier of the samples to the column and the detectors. The second term is stationary phase, where you always talk about column, how the column separate the unknown samples. Okay, so these few terms is very important. Okay, the separation depends on the reaction or interaction between the unknown samples, compounds, and the column itself. So the next session we will share about what are the HPLC components and what are the functions in very brief. Okay, so this is the modern look of the HPLC instrumentation. In PerkinElmer, we have LC300s as the latest model of HPLC. And in your lab with Dr. Audrey, probably, or Dr. Ali, we have the Flexa system still in range. And these two ranges would serve you different requirement of pressure. Okay, so you can see, first, there are a few bottles of solvent sitting up there. Actually, this is the solvent organizer. And we have the degasser inside this, mainly to remove the bubbles whenever the solvent being pumped into the system. Okay, why do we have a few bottles here is because the separation is based on different situation, or for example different polarity, different pH environment to the column. So we have to normally we have a few bottles here, okay, to create a gradient so that we create different environment to separate the compounds. And we have the Autosampler. To pump it on to entire system, we need to use a pump because we no longer use the gravity. And then at the same time, Autosample is where you accommodate your samples in the sample wires, okay? And also the column oven to accommodate the column, okay, with different temperatures. So there are many testing that is run under one room temperature, but there are also testing run under different temperature requirement, especially for those who want to mimic the biotechnology condition, like human body temperature 36.7 degrees, let's say. Then you might need a column oven to make sure the column is situated under the required temperature. And then finally, there are detectors to give you signals and peaks. Also, not to forget, LC300 comes with a waste management system where this system could organize the wiring and the tubings for your system so that the entire system looks organized and clean and neat. So this system is exhibit 2 detectors and 1 column oven with the basic unit of degasser, autosampler, and pump. Okay, but if you only need 1 detector, this is how it looks like. It could be stackable. Okay, it could be stackable. Still, the solvent rack, autosampler, and pump, and the detector could be situated up. Okay, if you have only 1 detector, you can do this. And also, you couldn't see the oven, column oven. It is because in Perkin-Elmer LC300 series, there is a column compartment, a small one, situated in the autosampler. So you can save some money from purchasing the column oven if you need. So this is the Flexa unit situated in your lab. Okay, same thing. Whatever model or whatever brand that you purchase, the basic unit should be the pump, autosampler, solvent manager, okay, and the detectors. Not to forget the column oven and injector as well. So most of the system nowadays are stackable and depend on how many detectors is required. You can arrange, the supplier could arrange the configuration for you based on your requirement. So this is a repeat slides to remind you what kind of samples could be injected into the LC. Okay, if you have the solid samples and you need to extract the samples into the appropriate solvent, then inject into the LC. But if you have a liquid, if the liquid is clean enough and the polarity is correct, okay, then you can inject directly. Else you might have to do some extraction before injection. Okay, one very important note to bring home: whether you have solid sample or liquid samples, after the sample treatment, it should not be precipitation or sediments being seen in the sample was, because this might clog the system if you have some sediments in the samples. So do some filtration before injecting into the HPLC. Okay, so we are going step by step, okay, to explain each part of the components in HPLC. First, we will explain the mobile phase, and then we will explain the degasser, Autosampler, pump, oven, and detectors. So before we operate the system, we need to select the mobile phase. So what kind of solvent that you should select for the testing? In pharmaceutical analysis, most of the time you already have a fixed method or from the literature or from the compliance method like US EPA or FDA method. So it already specify what kind of mobile phase. In general, the mobile phase should be soluble with the samples, and the solvent used might be HPLC grade, which is high purity, and the gas. So when you buy the HPLC grade water, HPLC grade methanol or acetonitrile, it shouldn't be refiltered anymore, because it is HPLC grade. For those analytical grade, even though high purity, but probably the sediment is there and it is not encouraged to be used in HPLC system. The next criteria of the solvent mobile phase is it should be not corrosive. So do not simply use any acid solution in the HPLC as there are many metal tubings involved. So it should be non-corrosive. And also the mobile phase that you chosen should be compatible with your detector. What do we mean compatible is if you are using the UV visible detector, then the mobile phase should not give you high absorbance in the UV system. So if you do so, this might interfere with your sample signal. Also, a low toxicity, reasonable price, and be it low viscosity all time. So the pumping of the mobile phase is smooth. This just now is the general characteristics of mobile phases, the requirement for HPLC, and you can also have to take care the common characteristic of mobile phase in terms of polarity. So as mentioned, if you are in the normal phase HPLC, then probably you need non-polar solvent like hexane and toluene. But if you are targeting the reverse phase components, particularly very hydrophobic, probably you need the reverse polarity solvent as mobile phase like water, methanol, and propanol. So these are the glance of polarity check of your solvent. And you can also choose the solvent based on the HPLC mode, as discussed earlier. Now the mobile phase is always having the reverse polarity to the target analyte. If you have hydrophilic compounds to be tested or non-polar compound to be tested, then probably your mobile phase must be a polar one so that it will not grab your target compound or having some competition in grabbing the compounds with the column. It just has to carry the compounds to the column itself and let the analyte to be interact with column. And in the column, same polarity will have the affinity to similar polarity in the analyte. We call it light dissolved light. So when you select the column, you also need to be careful with the polarity. So in short, when we choose the mobile phase for HPLC, we need to choose the reverse polarity compared to the characteristic of analyte itself. So the next would be, after the mobile phase carry our unknown samples into the column or stationary phase, what will happen? In these slides, this is the microscopic picture of the column itself. So what is inside the metal column of HPLC? This is just the example that we show in the dimension of 150 mm. And there are some thickness and pore size, particle size to be mentioned here. Okay, usually it is a very tiny kind of silica substrate, okay, like this, and you have a lot of pore size in between. So the sample and mobile phase is actually traveling between the slits, okay, or the pore, okay, between the silica substance. So what make the silica substance or substrate different to be packed in a column is the base is always the silica, but at the surface of the silica, we will pack the column with different compounds, which is vary in polarity or affinity. So in the reverse phase LC column, the most popular column that you heard of probably is the C18 column. So C18 is stand for 18 carbon attaching with hydrogen atoms, okay? But in total, there are 18 carbon atoms. So that will form a long chain hydrocarbon here with the carbon and hydrogen around. The total of carbon number is 18 being attached onto the surface of silica. And these C18 bondings are actually hydrophobic, okay, or nonpolar, and it will catch or absorb the nonpolar compound as well from your sample. So if you have a C18, then this end will be coated as C18 surface. And if you purchase C8 column, then the surface of the silicate compound, silicate phase will be coated with 8 carbon number of chain onto the surface. So this technology could serve different purpose of application or in HPLC, where when you coat it with different surface, you can absorb different characteristic of compound. And it is talking about different types of the absorption sites. Basically, it is falls into the surface chemistry and actually deserve for another lecture in depth. So for the general, you just remember this is a very small particle size and being packed with different surface, chemical surface, and being packed into a metal wall of column. So this is the illustration, okay? You can see the base is silica particle, okay? And the stationary phase like C18 or C8 compounds will be coated on the surface of silica particles. And at the same time, mobile phase is flowing all time, bringing the analyte going through the column. So whenever the analyte of higher polarity probably will not stay in the non-polar column so long and being eluted by the mobile phase to the detector at first. Whereas if you have a non-polar column, the non-polar compound will stay a little longer in the column because it likes or absorb longer in the column, okay, and elute to the detector later. Okay, so again, we would like to show this animation again. This is how they separate according to different condition in column. Okay, so different column phase will give you different sequence of elution, and also whether the slower rate or longer rate of elution to the detector. Okay, so you might also select the column according to your target compound characteristic, whether it is a high molecular weight or low molecular weight. Then out of it, it is a soluble one or organic soluble. Okay, so you can select a range of column according to the characteristic of target compound, and also according to the surface chemistry that we discussed just now, based on the characteristic of bonded phase. And you can choose now C18 or silica at the base, and probably you have C8 on top, or helix, or the amino surface could be used according to different different bonded phase chemistry in HPLC column. Again, this deserves another lectures. So today we will just want to deliver one message. Select the column appropriately to, this is the key of success in HPLC analysis. Okay. So the next one is the solvent manager. You will need a solvent manager to degas your solvent analytes, because whenever you have bubbles in the solvent or mobile phase, you will have inconsistent retention time in your chromatogram. So again, mobile phase for LC is very important. Okay, as you can see, the hardware like pump, column is only oven is on, and detectors is only it is a must, but the magic is done by the column and also the mobile phase itself. Okay, so based on the characteristic of your analyte, choose the phase of the LC phase. And when you choose normal phase or reverse phase, this will actually determine or decide for what kind of mobile phase to be used. So let's see what is the effect of mobile phase. Let's say you have different composition for the acetonitrile. You are using the water mixing with the acetonitrile, we call it ACN, in different composition. When you have 0% of ACN, 100% of water, you can see almost nothing is separating. You have a big hump at before the end of analysis, but there is no separation. Okay, and gradually, if you increase the composition of acetonitrile, you begin to see some peaks eluted. Okay, so this is what we want to show you. The correct composition of mobile phase will give you a different quality of chromatogram. Okay, so when you increase probably 20% ACN, you start to see some peak, but you can see that peak is slowly it is eluting at different rate when you increase the ACN. Okay, the higher the ACN composition here, the faster the elution of the target compound in this case. So the composition is very much important. Okay, and you can see this when you have 50% ACN and 50% water with the buffer of TFA, you have only 1, 2, 3 peaks, and the first peak is not well resolved. Probably these are two components, okay, being eluted at the same retention time. So to generate the suitable condition for this peak to be separated, okay, we would like to have one is lower, another one is eluting later. Okay, you can modify the ACN and water ratio. Okay, and now you have these two peaks separated. However, the last peak also started to see some changes. In the previous, you see only one peak here, but after you increase the water and decrease the ACN, now you can see a little separation here, which means the peak earlier here in 50% ACN is actually one peak. Okay, but it looks like one peak, but actually it contains two target compounds. So finally, when you fine-tune the method, fine-tune the composition of mobile phase, you have all the peaks separated. Okay, it is not 4 peaks, it is not 3 peaks, but actually the sample contain 5 components and now being separated well after fine-tune of composition in the mobile phase. So this is the effect of mobile phase, how the composition is affecting the quality of chromatogram. And also, other than the composition of mobile phase, sometimes to adjust the composition means in some cases, okay, not the ACN water, but maybe ion exchange in HPLC mode. Okay, it is some testing in HPLC is very sensitive to pH changes. Okay, in this case, when you change the pH, the sequence in the acidic condition, elution sequence is 1, 2, 3, 4. However, in the basic condition, pH 11, the sequence of the elution of every components could be upside down. And now you can see the component 4 or compound 4 is the second peak, whereas in the acidic condition, it should be last peak. Okay, so please take care if the method requesting you to adjust the pH precisely, usually you need to do very carefully so that you get the correct chromatogram. Okay. So we have been finished with the mobile phase and how it affect your analysis. And now, what about your Autosampler? So Autosampler, it is a device to let you put your samples. And inside here, there is a syringe to withdraw your sample. Usually in HPLC, you will need to inject around 5 to 50 microliter. So the benefit of having an autosampler is you can always inject such a small amount with very consistent withdrawal compared to the manual injection, because full of human errors, and from person to person, even in the same person itself, okay, we could be adjusting the syringe, withdrawing the volume of samples very uh, in a very big variation. So for a consistent area, consistent peak height, okay, we recommend you always to use the auto sampler. Okay, so the auto sampler, you can put your samples in a specific position, and it was numbered for every position here. So if you put at position 6, then in the software, you might key in my sample A is situated in vial number 6. And the system will instruct the hardware to withdraw the volume that you specified from vial position 6. That is the basic function of an autosampler, to let you put down your samples in a specific location so that you can specify that in the software. And in modern days, like LC300 now, this auto sampler could even help you in auto dilution. Okay, if you want to prepare a serial of dilution in standard from the low concentration to high concentration, you just instruct the software to do so, and the auto sampler has a mechanism to do for you. Okay, so dilution, serial dilution, sample preparation, the simple dilution of sample could be done with the modern autosampler nowadays. So the next one, we would like to talk about oven compartment. Okay, oven compartment is for you to maintain the temperature for the column. Okay, if you have specific temperature to be adhered, like body temperature or room temperature. At the same time, in LC300 of PerkinElmer, there is a small compartment in the oven for you to allocate the column up to 150 mm. And because of this autosampler could be temperature control. We allocate one column compartment here. This might save you to purchase another specific column oven. Okay, you can operate this HPLC with or without column oven, because there is a place for you to put the column in the auto sampler. Okay. Next is the solvent delivery platform. We call it pump. Okay, you can choose the correct pump limit for your HPLC. Okay, and there are other than the pressure difference, you can also choose whether you could put in 4 solvent or 2 solvent, because the category of pump is now separated to binary and quaternary solvent delivery. Okay, just a glance for this. Binary pump is just one pump, and you can put the maximum 2 solvent into the binary pump, whereas the quaternary pump, it is only one pump and become a cheaper option. They are using the proportioning valve to proportionate the solvent that the mobile phase that you allocate, maximum mobile phases to be allocated in quaternary pump is 4 because it has a proportionate WAF to adjust the ratio of each mobile phase, maximum up to 4 mobile phase. Okay. So this is a brief of the difference of both type of pump. Okay, especially when you purchase the HPLC, whether to go for binary pump or quaternary pump will be one of the factor to determine the cost. Okay, as you can see, binary pump has 2 pumps, each pump controlling 2 lines. Okay, but every time when you use the pump, you can only use 1 solvent line. So maximum of solvent here to operate for binary pump is 2 solvent base. But in quaternary pump, even though you have only 1 pump in operate, but the proportionate WAF will help you to adjust the ratio of each one. In one time, you can put in maximum 4 solvent as mobile phase. Okay. So when you do this, okay, the purpose of doing different ratio, okay, doing different ratio of solvent A and B or solvent A, B, C, D is actually to create different polarity environment or different pH environment. Okay, sometimes, okay, depend on test method. Okay, so that we could slow down some of the elution for some component, or we could fasten some of the elution to generate a different pace of each components to reach the detector. So for this, by doing this, we are able to separate the mixture into individual components to make them reach the detector at different time. So to achieve this objective, we sometimes need to adjust the mobile phase ratio. Okay, a little bit of A at beginning, a lot of C, solvent C at beginning, and then we adjust increase the proportionate of A solvent and lower down the C solvent and something like that. We call this as a gradient. Okay, a gradient. As you can see here, for example, I'm using methanol as a solvent A and the rest are water. Okay, at the beginning, 0 to 3 minutes, it is only 30% methanol. The rest is water. The flow rate will be consistent throughout the testing, and I'm using some wavelength to detect some target component. Okay, I expect let's say I'm expecting 1 peak to be eluted before 3 minutes under 30% of methanol, and the suitable wavelength is 330. For the rest of the time, I'm playing around the composition of methanol to create different situation of polarity to let different component to elute because each one of them has different preference in polarity, and I use a suitable wavelength. Okay, so this program, mobile phase program, we call it gradient. But if you are using only one composition from 0 to the end of analysis, that is isocratic program. Okay, so you can decide to increase, okay, to change the composition by proportionate rate or by the drastically changes of rate. Okay, this would be applicable in all the HPLC, the modern HPLC. The next is, let's see about the Peltier oven. Okay, it could control 5 to 90 degrees Celsius. So if you have specific temperature to be maintained for the column, okay, then you can purchase the external Peltier oven. It could accommodate longer column and up to 6 column compared to the Autosampler. Okay, and also the solvent manager to keep your tubing and wiring neat. Okay, and by pressing one button, you can turn off or turn on for the entire modules of HPLC. Finally, let's have a glance for the detectors. I'm so sorry, I think I'm behind of the time. Okay, these are the common detectors. Okay, basically you could segregate the detectors into a few types. Okay, UV-vis based, okay, UV visible based detectors, okay, or fluorescence or RI detectors. Okay, so UV visible detector, of course, the working principle is based on the UV absorption, and then RI detector is universal but less sensitive compared to the UV base. Okay, so the UV visible detector and the fluorescence detector are more sensitive compared to RI detector. So whenever you choose the detector, also consider the sensitivity and also whether it could be gradient setting or not. For example, RI is very sensitive about the pressure changes in hardware. Okay, so you couldn't set any gradient testing in RI detector. Whereas for the other two, the fluorescence and the UV based detectors, you could set the gradient to change the polarity situation in the system. Okay, so also your component must be reactive, okay, must be having some reaction absorption to the UV source or fluorescence source. Else it couldn't be detected and you have to choose other type of detectors. So I have some slides about how the UV detectors, the arrangement of the UV based detectors is you have a few types of UV detectors. Some you can only choose one wavelength for each analysis, and some you can choose a few wavelengths just for one injection. Okay, so whether multi wavelength or single wavelength UV absorbance. Okay, so it is based on the different arrangement of optical hardware inside the detectors and make you and this enable or disable the multi wavelength selections. You can also choose the PDA detectors where the unit could be scanned one range of wavelengths rather than only the selected one. Okay, so this PDA is going to scan a range of wavelength rather than despite of choosing the specific wavelength. Okay, and you have information and very useful for method development, especially you have really unknown sample to study. You do not know which wavelength to use for your sample, okay, or analyze. Then you can go for PDA. Okay, scan a range of wavelength and determine the maximum wavelength. And for this sample, I would say 250 nm would be the best wavelength for this analyte or samples. Okay, and also PDA could give you the 3D view. Okay, you have more information compared to only the basic UV detectors. Okay, so there are a few models. It is almost the same for all the suppliers. Okay, whether you could only choose one UV wavelength or multi wavelengths, or you have the PDA, also a UV based detector, but you could scan a range of wavelengths. Okay. Next is the RI detector. The working principle is based on reflective index. Okay. And finally, the fluorescence detector, okay, is using the principle of fluorescence. Okay, it is a type of electromagnetic spectroscopy. Okay, when the samples or analytes, okay, absorb enough energy from fluorescence light, okay, it will be excited and finally get down to the lower energy state for the stable state. Okay, and in the meantime, when the sample or analyte do so, it will release some lights, fluorescence lights, to be measured by the detector. Okay, so it is talking about different principles of working in different detectors. Okay, so in the summary, PerkinElmer could offer you from 6,000 psi to the mid-range of pressure limit, let's say 10,000 psi, up to the maximum 18,000 psi. So these are the range of HPLC that is available in our range. Okay, so far this 18,000 psi is the highest range of in the market. Okay, and when you select different column dimension, it will give you different back pressure. Okay, nowadays the packing material of the column is getting smaller and smaller, and this means the back pressure is increased. So if you have only 6,000 psi HPLC, probably there is a limit of column selection, and this will also result the limitation in the testing that you could do with the HPLC. Basically, the higher the pressure is, the less limitation you are facing in method setup. Okay, but most of the time, if based on the budget and the application, the mid range like 10,000 psi HPLC already could cater for most of the testing for pharmaceutical. Okay, not to forget whether the software is easy to use or not. Okay, those days the software in HPLC or chromatography system is quite you need to have a lot of knowledge to tabulate the peak, to integrate the peak, to set the parameter on how to integrate, how to identify, and thus calibrate the unknown. Okay, so the requirement is, okay, the flow should be easy to understand and easy to manage and operate. Also, not to forget, okay, the software should be integrated with 21 CFR compliance, okay, in pharmaceutical line. Okay, so Simplicity Chrome for LC300 could meet that. Okay, so thanks for that. And next, I would like to share maybe another 10 minutes to share with you how to apply the HPLC in the pharmaceutical line. And in today's presentation and examples, I would like to take a type of traditional medicine and drugs to be identified qualitatively, okay, with the HPLC, and then quantitate it. Finally, after the quantitation, have to make sure whether this compliance to the FDA requirement or the method requirement. Okay, so the example that I would like to share with you is the analysis in Lonicerial japonica floss by the UV detector in HPLC. So this is a kind of herbs. First, we will characterize the compounds that is required in the pharmacopoeia method. Okay, it is a Chinese medicine in a herb. Okay, so it is very popular for the inflammation and infectious disease medicine. Okay, so in the manufacturing process, okay, we have to take care of the quality control. Okay, first identify the peaks and then determine the amount or quantitate the amount or assay of this compound. Okay, so in Chinese pharmacopoeia method, it states that in these samples, 7 components must be detected. Okay, 6 of them is identified in these examples, and in the method of pharmacopoeia, it has an acceptance and tolerance limit after we identify it. Okay, so we have to calculate relative retention time in relation to a reference peak. Okay, that means to say how close between 7 peaks to each other. Okay. These are the 6 components that are detected in the HPLC method. Okay, by using the Perkinamar LC300 system with the C18 column. Okay, so these are the chlorogenic acid, which is quite famous. Okay, and the rest, okay, are the components listed in the Chinese pharmacopoeia. Okay, first we are doing the qualification or identification. Okay, so this is the experimental sample preparation. Okay, first prepare the standard solution. Okay, dissolve the 400 microgram per mil to a series of different solution. Okay, and then treat the sample. Okay, basically this is just to extract with 75% of methanol solution. Okay, sonicate 30 minutes and then filter through the syringe filter of 0.22 micrometer. Okay, after filtration, then we can inject the sample and standard to the HPLC. So this sample will be compared against the standard solution. The standard solution here is chlorogenic acid. Now, this is the LC parameters. Usually the method will specify how to set the method. And sometimes the operator or chemist or pharmacist need to fine-tune the gradient program of gradient mobile phase, okay, to have a better separation. Okay. So this is the results. Okay. The single compound here is the chlorogenic acid, okay, from the standard, and the below chromatogram is from the herbs, okay. So normally this kind of natural samples is having a little bit noise area, okay. The baseline is having some noise and interference, but in this example, the chlorogenic acid is separating very well, and also the other 6 components have been identified, and then the software will calculate the peak symmetry, how symmetry is the peak, okay, whether it is compliance to the Chinese pharmacopoeia requirement or not. We call it system suitability specification, okay. Probably you are not you are quite familiar with this kind of terms, okay. And also the software will calculate what is the plate number of the column, okay. So after analysis, we have all in, and the retention time, retention time means how far or how close is both peaks. So the system is going to calculate everything here, okay, and we find it system is working well and compliance to the Chinese pharmacopoeia requirement. Okay, so this is the summary of the RRT, relative retention time. So this is to calculate how far to each other, referring to the Ca, chlorogenic acid. Okay, so all of this has been listed in Chinese pharmacopoeia. Okay, and what is the deviation from the stated reference peak? Okay, is listed. And these are the accepted RRT. Okay, what is the range and what is the results? Basically, plus minus 10% of the value could be accepted. So this is fine. After you've done the identification or qualitative analysis, now is the time for us to quantify the chlorogenic acid. Okay, so these herbs is actually, the chlorogenic acid is actually popular in making the cough medicine, okay, but it is falls under the traditional throat reliever granules. It is a traditional Chinese formulated medicine. It is not the clinic kind of Western medicals, okay. So the produce of these products is required to adhere to some QC or quality control process based on the Chinese pharmacopoeia, okay. So the limit is chlorogenic acid could not be more than, okay, could not be less than 3 milligram per bag. Okay, every bag here of the medicine must be more than 3 milligram. Okay, so the total weight is 4 gram. Okay, so our target compound is chlorogenic acid now. Same thing, we are setting up the instrument by a method like just now and using the column, recommended column, C18. And then we prepare the sample and standard. This time, we no longer prepare only one standard, but we need to prepare a series of standard with different concentration, because now we have to quantitate it. Okay, just now only one standard we just identify. Okay, now we already know how to dilute the standard. Now sample preparation is still the same. Okay, basically it's an extraction by the methanol solution and sonicated, finally filtered. Okay, after filtration, you can inject into the HPLC, and this is the result. Okay, so standard solution, okay, shows a single chlorogenic acid, okay, whereas the sample itself, okay, the finished finished medicine itself showing a few peaks. And now with the sample and standard comparison, you can identify where is the chlorogenic acid situated. So the peak is around 9 minutes here. Okay, same thing. The software will calculate the peak symmetry and the theoretical plate number for you. Okay, you don't have to calculate by yourself. Okay, and then they also inject a blank solution where only the mobile phase in place, and also injecting only the 50% methanol that is being used for sample prep. And they found there's no interference, nothing eluted, means the solvent that they choose, the column that they choose, and also the gradient program in HPLC are excellent for the separation in this case. Okay, so this is the repeated injection for chlorogenic acid, 5 times injection, getting the similar area, and the repeatability is very well. Okay, basically you get not more than 0.5%, just 0.2%. Okay, and then also the retention time. Why do we need to calculate the retention time repeatability? Is we want to ensure every time the chlorogenic acid present, it would be show up at the same retention time. So this is also the requirement for Chinese pharmacopoeia. Okay, if the retention time is changing all time, up, up and down, front and behind, then it is quite difficult to identify the chlorogenic acid or any other targeted component. Okay, so finally we plot a graph. Okay, by using the area under the peak. Okay, with different concentration, we finally plot a graph of the area versus the concentration or amount, and we draw a calibration curve. Okay, and then inject the unknown sample. So this is formed by 2 microgram to 80 microgram of standard, giving you the different area. Basically, when the concentration increase, the area will be also increased. It is proportionate. Okay, and we inject the unknown sample. Finally, we quantitate, we get the area of chlorogenic acid in the sample. For example, here, okay, it's 2,500 area, and then we touch the calibration curve and read the acid X, and now probably is 50 microgram per ml. This is the example, and we use the technique to quantitate how much is the chlorogenic acid in every bag of finished product in Chinese medicine. Okay, so the the result is 3 milligram per bag. Okay, so this is the example of how are we going to use the HPLC in pharmaceutical products. Okay, PerkinElmer could supply you from the hardware of HPLC to the consumables like columns, and also we have the methods. We have a very well software to do this, and most importantly, the service and the training is done by one PerkinElmer source. Okay, rather than asking for a few suppliers source. Okay, I hope this sharing is bringing some idea for you of what the HPLC can do in pharmaceutical line. So that's all of my sharing today. Dr. Ali, I will hand over to you. Thank you. Um, I'm sorry, Dr. Ali. In fact, I could not hear you. Yeah.
Sorry. There was a little bit of glitch. Thank you so much, Ms. Chong. That was a brilliant presentation, and I have learned a lot. And I believe our viewers have learned a lot at the same time. It was a very good, informative presentation, and there's a lot of interesting questions which have flooded up during and by the end of the presentation, one of which is over here by Miss Audrey Young, and she is saying, hi, Miss Chong. Great talk. I need some advice. Can I know what should I do? Restart our HPLC that has been idle for 12 months. Should I flush the system with water or solvent? Yes, what should we do?
Yeah. It is just like every time when you start the LC, whether 1 week of not using it or 1 year of not using it is pretty similar. So for myself, I will start up the system and without the column at first. Without the column means you detach the column, okay,
Mm,
and you divert the outlet or inlet of the column tubing into a beaker. Yeah, you can start with the water at first, or even some 70% acetyl nitrite or 70% methanol, okay,
mm,
30% water. You set an
mm,
isocratic program or gradient program, doesn't matter, because now the column is not here. Okay, you can also 100% water flushing and slowly adjust to 30% of ACN or methanol. Okay, and then you take a extreme one, 70% of organic solvent. Okay,
all
after
right.
that, yeah, this is just to check at whether the system is clogging. If not, then now you can stop the flow, connect the column, and you adjust 70% organic solvent. Start with a slower rate, like zero point 0.1 ml per minute, slowly increase to zero point 0.5, and you see a tiny drop from the column eluted out. You don't have to direct to detector. Same, you can put the column or with the tubing and direct it to a waste beaker. Okay. When you see there is a drop of solvent, then you can slowly increase the flow rate to the experimental flow rate, like 1 or 2 ml per minute, and you flush about 20 to 50 times of column volumes. And if you don't want to care about that, you can even leave it 30 minutes to 1 hour just to rinse out the column, activate the column. Yeah. And you are good to go. Yeah.
That's amazing. I hope the Audrey Young was writing that down somewhere. Now, the next one, there's a lot of questions. I'm sorry, I'm going to bother you a lot of questions here. Miss Shahira Wahab. Hi, Miss Chong. How to differentiate different compounds with same UV spectra and retention time? Very technical.
Now, it's a good question. So every analysis technique has the limitation in practical. So
Mm,
as you can see, what we can produce from the HPLC is retention time and also the UV absorbance, for example. Okay. So if there are two compounds that is using the same wavelength and eluted at the same retention time, there is no way that you can make any conclusion about the amount of each one. So what we can do is probably we will try to find different wavelength for both. Okay. Probably
mhm,
there is one wavelength that is only applicable to one of them. Else, if you have option, you really need to select other technique like
mm,
different detectors, different working principles, including the mass spectrometer. If you
mm,
have the mass spectrometer, you can separate by using the software by keying in the different molecular weight.
mm.
Then the chromatogram should be able to separate by that. Yeah.
All right. Then there is another question from Miss Peachy Lim. Hi, Miss Chong. Thank you for your informative talk. Can you give more info? How effective does HPLC in separating chiral compounds? People are asking
Mm,
about their PhD and master questions from you, Miss Chong.
yeah, yeah. I'll try this. Yeah. The separation, you have problem when you have two very similar isomers, whether isomers or similar compound structures. Okay,
Mm,
that is really difficult to be separated because they might have similar polarity or similar absorbance to the column phase. Okay, again, when you put in the mass spectrometer, now their molecular weight is same because they are isomer. Okay, probably the
mm,
geometry of molecular structure is different, but the weight is same. So again, this comes to the limitation of uh, technique, analysis
mm,
technique. But nowadays, HPLC technique, they develop some chiral columns. So
mm,
this would be something to do with the column selection. They are now developing some useful chiral column for this kind of compound. So do not only limit yourself in C18 column. Sometimes by changing a correct chemical surface, you could separate the column well. So separate the analytes well. Yeah.
mm. That's a
But
very good answer.
yeah, column selection is part of the try and error or method development as well. Yeah.
Mm. All
So the
right.
pro
Oh, please go on. Please go on.
yep.
All right. So there is another question, and this one is related to the first one, which you answer. Is it advisable to use warm water to flush the system?
Not necessary. Not necessary. Yeah.
Mm,
Yeah. You do not have to use the warm water to flush the system because there is some seal or O-ring in the system that
mhm,
we do not very sure of the temperature tolerance, and this is not quite safe for the system. Yeah.
mm. All right. Okay, two more. Okay, so Miss Nick Atira Rashida. Hi, Miss Chong. Thank you for your sharing. Just one question: how do I overcome if my sample has poor solubility in polar solvents? Solubility issue.
You okay? Then you might need to change the mobile phase. Yeah.
Oh,
Yeah.
I
So
understand.
we could not change the natural characteristic of the compound. We
Yes.
are not supposed to do so. So do not force the compound.
Mm,
You will never win. This is nature of the compound itself. So
true.
why not you try some non-polar solvent? Yeah,
Mm,
and then be careful with that. When you turn that to the normal phase, then you might need to consult the supplier whether you could do that with the system or not. Some system is convertible between normal phase and reverse phase, means the changes of solvent in polarity. Yeah, some system is limited to do so. Yeah.
mm,
So better to consult the supplier. Yeah, because again, there are some seal and O-ring inside, probably will be melted if you change to non-polar solvent. Things
mm,
like that,
mm.
you need to take care. Yeah. And also when you change a solvent type, the pressure in the system will also change. Yeah.
Oh,
Yes.
okay.
So you can observe by pumping 100% of water and pumping 50% of methanol with water, you can observe the pressure change. Yeah,
Mm,
so
mm,
all
mm.
this you could consult the engineer of the suppliers. Yeah.
Absolutely correct. Now, I think that would be our last question. Thank you so much, Miss Chong. And honestly, I want to compliment this. We have never received this much questions from our viewers. You're the first one, and I believe the interest was high, and that's why the questions were higher as well. Thank you one more time, and I
Thank
believe you
you.
had a good time.
Yeah, thank you, Dr. Ali. This is my first stream, my live stream, and I would be very happy. I'm very happy to have it with Masa University. yeah, I hope to see you in future again. Yeah, it seems that
Same.
we have a lot of HPLC users. Yeah, so please keep in touch with us.
Yes,
Yeah,
please. Yes, Miss Chong, thank you so much. And thank you for all the viewers who have joined us. Don't worry, we will keep Miss Chong on board. If you have any more questions, please keep them alive for the next session. Thank you, everyone. Enjoy yourself and stay safe. Thank you.
thank you.